viewer, today I will discuss about allosteric enzyme principle and mechanism. I will talk about the regulatory enzyme, definition, types and example, then allosteric enzyme definition, mechanism, properties, features, characteristics, models, covalently modulated or modified enzyme as well as protein processing. So, enzymes can be regulated by other molecules. So, it can increase or reduce its activity. The molecules which increase the activity are called activators, and the molecules which reduce or decrease its activity, they are called inhibitors. All enzymes exhibit various features depending upon the cellular substrate content, intracellular pH, temperature, ambience, etc. Regulatory enzymes. Regulatory enzymes are mainly two types. Number one, allosteric enzyme. Number two, covalently modulated enzyme. In case of allosteric enzyme, which is modulated through the non-covalent binding of the modulator other than active site. And in case of covalently modulated enzyme, the enzyme is interconverted between active and inactive forms by action of other enzymes. These two enzymes, they are responsive to alter the metabolic status of the cell, and in both the cases we can find they act for short time span. In case of allosteric enzyme within second, in case of covalently regulated enzyme within minutes. Allosteric enzyme. The name was proposed by Monod, Shangu and Jacob of Pasteur Institute, Paris. Enzyme regulation. So, when I will discuss about the enzyme regulation, it will include allosteric control, covalent modification, protein processing or zymogen activation, stimulation and inhibition by control proteins, genetic control by repressor or activator. Allosteric control or regulation. Allosteric control refers to a type of enzyme regulation involving the binding of a substrate or a non-substrate molecule known as allosteric effector at location on the enzyme other than catalytic or active site. So, at another place or other site is referred, it is called allo site. The allosteric site is a site which actually accommodates small regulatory molecule which can activate or inhibit the activity of the catalytic site. The binding of allosteric effector is a general, non-covalent and usually reversible. Allosteric enzyme principle. So, I have already told allo means other site. So, the activity of the enzymes are regulated by binding of specific ligand, effector, modulator. There are so many different names. So, all those molecules and regulator can regulate the activity of the enzymes. For the enzymes, usually two or more structural shapes are there which have differential ability to bind to the substrate. Two major groups of the allosteric enzymes are there. One group is regulated by changing the affinity or by changing the K-Michaelis Menten for the substrate, for one substrate specifically, keeping its maximum rate or velocity somehow constant, that is Vmax is constant, fine. So, they are known as K types of enzymes. The second group does not demonstrate the significant changes in the substrate affinity, but has large changes in the maximum rate or Vmax changes. So, they are usually referred M type of enzymes or V types of enzymes. Now, so what I have told you, in case of regulatory enzyme, there are multi subunits are present, fine. So, when at the regulator site modulator or regulator binds, so somehow this binding is transduced to the catalytic site. So, we can find at the catalytic site the substrate binding is getting changed. So, somehow the another kind of active enzyme substrate complex is produced. So, from the less active enzyme, more active enzyme is produced if the modulator is the enhancer or activator. Allosteric enzyme characteristics. Allosteric enzymes undergo conformational changes in response to modulator binding. Based on modulation, they can be subgrouped. Homotropic allosteric enzyme. Only substrate or product of the enzyme itself act as modulator. The next type is heterotropic allosteric enzyme. Here the modulator is other than substrate or the immediate product. Allosteric site can be polyvalent or monovalent. That must be specific for specific types of modulators. Polyvalent means they respond to two or more specific modulator at different specific sites. So they can accommodate different types of modulator at different binding site, but the sites are specific. I will talk about the cooperativity, I will talk about the kinetics, but we have to keep in mind that allosteric enzyme, they follow the sigmoid kinetics. Cooperativity, that is binding of one substrate, enhances or decreases binding of other substrate, and it follows sigmoid kinetics behavior. Generally, it reflects the cooperative interaction between the protein subunits, catalytic subunit, regulatory subunits, etc. Sigmoid kinetics behavior generally reflects cooperative interactions between protein catalytic as well as regulatory subunits. Changes in the structure of one subunit is translated into structural changes of adjacent subunits. An effect is mediated via non-covalent interaction at the interface between the subunits. So, characteristics of sigmoid kinetics is that small changes in the concentration of a modulator can be associated with large changes of activity, that is V0, rate of reaction. So, we can find the effect of positive as well as negative modulator, and we can find the positive as well as negative cooperativity. I will discuss about that. So, here we can find the apparent changes in the Km value. Okay, so that is the substrate affinity. So, we can find K series of enzymes. We can find for the negative modulator, the Km will be increased, and we can also find where the Vmax will be changed, the M series of enzymes. Biological significance. Not all the allosteric enzymes, they belong to either K or M. They may belong to in between them. Regulatory proteins, if we follow the plot of initial velocity versus substrate concentration, that is biologically significant only at lower substrate concentration, since the concentration of the metabolites in the cell are far below saturation levels of enzyme acting on them. Features. Allosteric enzymes, they act through reversible non-covalent binding to the regulators. They may be the small molecules, maybe substrate or cofactors, so they actually modulate the activity. Sometimes we may follow the irreversible covalent modification. So what is going on? Sometimes some separate types of regulatory proteins are present. They regulate the activity of the regulatory proteins. So these regulatory proteins are the pacemaker of the cell. They can regulate, either they can slow or they can make the faster of the reactions when the substrate concentration or the metabolite concentration vary within the cell. They have the sense or signal, yes, all those factors are varying. By response to them, they actually change the reaction rate within the cell. So they are the pacemaker. Why they are called the regulatory enzymes? Because they are able to regulate the metabolic pathway. On other hand, they are regulated by other enzymes, other metabolites, etc. So that they are called the regulatory enzymes. Properties of the allosteric enzyme. As we know, allosteric enzyme is a type of regulatory enzymes. So allosteric enzymes undergo conformational changes. The modulators of allosteric enzymes may be stimulatory or inhibitory. Regulatory enzymes for which substrate and modulators are identical are known as homotropic enzymes, or effector is called the homotropic effector. Heterotropic enzymes have substrate other than the modulator, other than the same substrate of the enzyme. Allosteric enzymes are generally larger and more complex. The properties are significantly different from the normal enzymes. Allosteric enzymes have one or more regulatory sites other than active site for binding with the modulator. As the active site of the enzyme is specific for the substrate, similarly each regulatory site is specific for each and every modulator. So what I have told? The complex, the regulatory enzymes or allosteric enzymes, they have larger structure, more complex structure and more difficult to purify. All are usually oligomeric, having more than one subunit or polypeptide chains, usually even in number. It shows anomalous properties. Some are stable at room temperature as well as in vivo temperature, but unstable at 0 degree centigrade. They exhibit a typical dependence of reaction velocity on substrate concentration. Inhibition of them does not follow prototype competitive inhibition, non-competitive inhibition or uncompetitive inhibition. Many allosteric enzymes are mixed homotropic heterotropic type where both substrate and other metabolite function as modulator. Desensitization. Sometimes with the exposure to gentle heat or urea can do the chemical modification of the modulator sites. So, it loses sensitivity to modulators. So, what we can find that the capacity of allosteric enzyme for activation or inhibition by specific modulators can be abolished without damaging the catalytic activity. So, what is going on? The regulator sites are detached or desensitized from the catalytic site. So, catalytic site is now become free of the regulator sites. So, it shows the Michaelis-Menten kinetics. Mutation synthesizes some defective allosteric enzyme which is insensitive to modulator for replacement of some conserved normal amino acids. Mechanism of allosteric enzyme. Allosteric enzymes that I have already mentioned do not follow normal Michaelis-Menten kinetics. This is because allosteric enzymes have multiple active sites. The multiple active sites exhibits the property of cooperativity, where the binding of active site affects the affinity of other active sites on the enzymes. Allosteric enzymes are influenced by substrate concentration. There are two conformational forms of allosteric enzyme, that is tight capital T or taut and relaxed capital R forms, which exist in equilibrium. Only at high concentrations of substrate, more enzymes are found in the R state. When there is insufficient amount of the substrate, the enzymes are found in T state. So, the T and R state equilibrium depends on the concentration of the substrate as well as modulator itself. Physiological significance of the allosteric enzymes. Allosteric enzymes are regulated by other molecules. This is seen when the molecules of 2,3-biphosphoglycerate and carbon dioxide modulates the binding affinity of hemoglobin to oxygen. Hemoglobin is an example of allosteric protein. 2,3-biphosphoglycerate reduces the binding affinity of oxygen to hemoglobin by stabilizing T state. Lowering the pH from the physiological pH 7.4 to 7.2, that is the pH in muscles and tissues, favors the release of oxygen from the hemoglobin. So, hemoglobin is more likely to release oxygen when it finds more and more carbon dioxide in the metabolizing tissues in the body. Now, I will start the allosteric enzyme kinetics. I will discuss about its deviation from the Michaelis-Menten kinetics, example of allosteric enzyme and the models of allosteric enzyme regulation. Deviation from Michaelis-Menten kinetics. The relationship between V0, that is rate of enzymatic reaction, and substrate concentration in allosteric enzyme is quite different from that of Michaelis-Menten kinetics. They do exhibit saturation kinetics with the substrate when substrate concentration is sufficiently high. But for some allosteric enzyme, plots of V0 versus concentration of S produce a sigmoid saturation curve for cooperativity, rather than the hyperbolic or rectangular hyperbolic curve of non-regulatory normal enzyme. In allosteric enzymes, the binding of substrate to one active site of enzyme can affect the properties of other active sites, may enhance the binding of subsequent substrate molecule to the other substrate sites in the same enzyme molecule. The binding of substrate to one active site of the enzyme facilitates substrate binding to the other active sites. And this cooperativity results in the sigmoidal plot of V0 versus concentration of S. On the sigmoid saturation curve, the value of concentration of S at half maximal V0 can't be designated as Km, Michaelis-Menten constant, because the enzyme does not follow the hyperbolic Michaelis-Menten relationship. So, it is called apparent Km for the substrate, or sometimes is designated as S.5 or K 0.5. The plots. The velocity versus concentration of ACE curve is sigmoidal here in case of allosteric enzyme, but for normal enzyme it is rectangular hyperbolic curve. In case of V type of enzyme, we can follow differential Vmax where K.5 is unaltered, but in case of K types of enzyme, differential K.5 are found though they are acquiring or reaching to the proper Vmax value. Homotropic allosteric enzymes generally are multi-subunit proteins and the same binding site on each subunit functions as both the active site and regulatory site. The substrate acts as a positive modulator as binding of one molecule of substrate to one binding site alters the enzyme's conformation and enhances the binding of subsequent substrate molecules. It shows positive cooperativity, that is, binding of one substrate molecule at one site enhances the binding of subsequent molecules at other sites. Sigmoid kinetics involves small changes in the concentration of a modulator can be associated with the large changes in the activity or V0 values. In heterotropic allosteric enzymes, the modulators are metabolites other than the normal substrate or the direct product, and therefore the substrate saturation curve changes according to modulator. Some heterotropic allosteric enzymes, in some cases, they are modulated via activating modulators, while some have inhibitory modulators, and some have both inhibitory as well as activating modulators. An activator or positive modulator may cause the curve to become more nearly hyperbolic with a decrease in K.5 but no changes in Vmax, resulting in an increased reaction velocity at a fixed substrate concentration. A negative modulator or an inhibitor may produce a more sigmoid substrate saturation curve with an increase in K.5. Negative cooperativity flattens the plot of initial velocity versus substrate concentration. Enzyme is far less sensitive to small changes in substrate concentration and approaches saturation more slowly. The enzyme aspartate transcarbamylase or ATCase is an allosteric enzyme which catalyzes the first step of the synthesis of CTP. Carbamoyl phosphate as well as aspartate condense into N-carbamoyl aspartate, which is actually the first reaction metabolites for the CTP biosynthesis of the total reactions. And CTP, the end product of the reaction, is acting as negative modulator, and CDP and CMP have no effect on the enzyme. ATP is positive modulator. We know from CTP we can find the interconversion to ATP as well. ATP is acting as positive modulator, which reverses the inhibitory effect of CTP. CTP increases Km, but ATP reduces it. So, from this reaction, we can find how carbamoyl phosphate and aspartate joins by the help of ATCase to produce carbamoyl aspartate, the first reaction in the CTP biosynthesis pathway. About the enzyme. This first enzyme was isolated and studied by Gerhardt, Purdy and Skachman. The molecular weight is 31 into 10 to the power 4 Dalton and can be dissociated with mercurial reagents to give 2 identical catalytic subunit and 3 identical regulatory subunits. Each catalytic subunit is 100,000 molecular weight and contains 3 polypeptide chains called C chains and each binding site for substrate aspartate and not sensitive to the modulator. Each of 3 regulatory subunits have no catalytic activity but contains 2 R chains having molecular weight 17,000. 1 atom of zinc is bound to each R chain. Each regulatory subunit binds 2 molecules of CTP. When dissociated 2 catalytic subunits, we can find that if catalytic subunit is dissociated from the regulatory subunit, catalytic subunit is no more regulated by the regulatory subunits. So, there are 2 conformational form. One is intact enzyme, active form, as well as it is reversible with the inactive forms when they are getting dissociated either or when they are conformationally changed to other form. By binding to the CTP, the active conformation is transformed to the inactive version, but by binding with ATP, once again it goes back to the original active conformation. For inhibition or activation by CTP or ATP to occur, C and R chain subunit must be associated. Physical association of oligomeric subunits are very much needed. And this signal of binding to the modulator is transmitted via conformational change, non-covalent interaction. CTP binds to R subunit, cooperative transformation happens from active to inactive conformation. So this is a representative enzyme. Some single polypeptide enzymes are also allosteric in nature, but they must have different sites for action of catalytic as well as regulatory actions. Allosteric enzymes exhibit sigmoidal kinetics rather than Michaelis-Menten kinetics. This is because the enzyme oscillates between two distinct conformation states, much like hemoglobin. The T state is characterized by low substrate affinity and low catalytic activity. The R state conformation is characterized by an increase in substrate concentration, simultaneously increasing in reactivity of ATCase. Allosteric regulation is species specific. E. coli and other bacteria, they are inhibited or activated by CTP and ATP respectively, but animal tissues, they are not affected by these two. Homologous allosteric enzymes may have different modulators. Allosteric regulation of ATCase that I have already told, ATP can also bind to the regulatory site of ATCase, but it increases the activity. Heterotropic effect, as ATP and CTP, they are not directly substrate or product of the enzyme, so they are actually heterotropic regulator. The heterotropic positive modulator is ATP and heterotropic negative modulator is CTP. The binding of CTP stabilizes the T state and decreases substrate affinity. The cooperativity, yes, the rate of product formation, that is N-carbamoyl aspartate, increase as the concentration of aspartate increases. In cooperativity, binding at one site increases or decreases the binding in other site of the enzyme. This is due to conformational changes of the neighboring subunit, it actually reduces. So this curve shows for CTP binding, we can find which is acting as negative modulator. This curve shows that apparent Km is getting increased, but for ATP binding we can find apparent Km is getting decreased. That means substrate affinity is getting increased from the normal level, and these two cooperative curves actually manifest them. The sigmoidal dependence of V0 versus concentration S reflects subunit cooperativity. Most allosteric effects can be explained by the two models. One is concerted or symmetry model or MWC model, which actually proposed by Monod, Wymo and Shengu, or by the sequential model described by Coastland, Nemethi and Fimmer. Both postulate that enzyme subunit exists one or two conformations, that is tense T or relaxed R, and that relaxed subunits bind substrate more readily than those in the tense state. The two models differ most in their assumptions about subunit interaction and the pre-existence of both the states. The first one, the mechanism of regulatory activity. Sequential model proposed by Coastland, subunits undergo individual sequential changes in conformation. Intermediate conformational states are found, but in case of symmetry model proposed by Monod, the binding of the first substrate increases the tendency of remaining subunits to undergo transition to high affinity from all or none effect. So, what we can find in case of concerted model, by binding to first substrate or modulator, all the subunits are getting changed together. But in case of sequential model, there are some intermediate transitional conformational change. Sequentially they are getting changed from either high to low affinity or low to high affinity. Allosteric enzyme in metabolism can control their metabolic activity, as we can find in case of feedback inhibition. The end product of the multi-subunit or the multi-step enzymatic process can inhibit the first reaction, which is called the committing reaction. In case of isoleucine production from the threonine, we can find the first enzyme is threonine dehydratase or threonine deaminase, which converts threonine to alpha ketobutyrate. Here the first enzyme is inhibited by the last product, isoleucine. Okay, isoleucine is acting here as heterotropic allosteric negative modulator. So in this way, whenever the concentration of the end product exceeds according to the cell's requirement, it goes and binds to the first committing reaction enzyme and it stops. So in this way the metabolism is actually regulated via such kind of non-covalent and reversible binding of the end product. It is also called end product inhibition. So feedback inhibition means the end product is inhibiting the first enzyme of the multi-enzyme reaction. Feed forward stimulation, sometimes precursor can stimulate the first enzyme or activate to run a particular metabolic reaction. We can also find some branch point inhibition as well. Let us take an example for pyruvate kinase, which actually converts phosphoenol pyruvate and ADP to ATP and pyruvate. Here the homotropic negative allosteric modulator is ATP and heterotropic feed forward positive stimulator is fructose 1,6 biphosphate of the glycolysis pathway. Covalent modification. So apart from these allosteric enzymes, some covalent modification of enzyme can be done either by phosphorylation, adenylation or uridyllation, ADP ribosylation and methylation. Regulatory enzymes undergo reversible covalent modification. The example is glycogen phosphorylase. It actually hydrolyzes or breaks down the glycogen present in muscle and liver. When the signal comes as epinephrine hormone, we know that the glycogen phosphorylase B is more inactive because there are two forms, glycogen phosphorylase A active form, glycogen phosphorylase B inactive form. Now 2 units of phosphorylase B having special serine residues, they are getting phosphorylated by phosphorylase kinase, are getting transformed to active phosphorylase A form. Then only it becomes able to break down glycogen and to form glucose 1-phosphate and reduced residue of glycogen. We can find by the presence of phosphorylase phosphatase, all those phosphate residues can be removed from the active phosphorylase A and once again it goes back to the original inactive version of phosphorylase B. So we can find the amplification cascade. One signal can actually activate more and more phosphorylase kinase, which in turn activates the phosphorylase from the inactive form to the active form. Protein processing or zymogen activation. So within the cells, some zymogens are getting synthesized which do not act upon the substrate. When only they are secreted out from the cells to the gastric juice, then only just by hydrolysis, by the acidic condition or by other enzymes, they become activated by exposure of the active sites. So that is irreversible process of enzyme modification. In case of covalent modification, what we have found, 2 sets of enzyme is there. One is activating them and phosphorylase phosphatase, which actually help to make it back to the original concentration, original form just by removal of the phosphate group. But here the pathway is irreversible one. Say for example, pepsinogen, by the action of acidic pH as well as pepsin, it is converted to pepsin by the removal of peptides. Trypsinogen by the help of enterokinase is converted to trypsin by the removal of hexapeptide. Chymotrypsinogen with the help of trypsin, actually 2 peptides are getting removed and original, the active chymotrypsin is found. So first it is getting converted to pi chymotrypsin from chymotrypsinogen, then active alpha chymotrypsin. Stimulation and inhibition by control proteins. Say for example, protein kinase A, when the catalytic and regulatory subunits are associated together, they are inactive. But when only the cyclic AMP comes and sits over the regulatory subunits, the catalytic subunits are dissociated and protein kinase A becomes activated. The genetic control can be done either by repressor or activators. We can find in case of lac-operon, when repressor sits over the operator, it inhibits RNA polymerase to transcribe LAC-ZYA gene. But when only the allolactose or inducer binds to the repressor, it helps to fall off from the operator and helps RNA polymerase to transcribe them. Once again, activator action can be found in case of maltose operator, where we can find RNA polymerase is unable to transcribe them, but maltose activator protein, when it binds with the inducer maltose, it actually pushes RNA polymerase to continue the transcription. So I have already talked about the regulation or the regulatory enzyme, allosteric enzyme kinetics, mechanism, property, features, characteristics and models, as well as covalently modulated and activated enzymes. Thank you.